Something in you has been holding on today. A worry you couldn't quite set down. A name or a number that slipped just when you needed it. A quiet fear that you are not as sharp as you once were. That fear has a name in neuroscience. The brain does not decline passively. It rewires. Every night, at the hours when the rest of the world has gone dark. It runs a process so complex and so deliberate that scientists spent decades not knowing it was happening at all. And here is the part most people miss. 3 in the morning, the hour you may have been staring at the ceiling, is precisely when that process peaks. Welcome to Sleepy Science. Tonight begins where the day's noise ends. Close your eyes. Breathe. Your brain has been waiting for this. Not to rest, but to work. Let the thinking soften. And now we begin. Your brain never truly goes offline. While you sleep, it enters one of its most active states. Not resting, but reorganizing. Neuroscientists discovered in the 1990s that neurons fire in coordinated patterns during sleep that closely resemble the firing patterns from earlier in the day, replaying events at compressed speed. This process, called memory consolidation, is the brain's way of deciding what to keep, what to strengthen, and what to release. It does not wait for you to ask. The moment you fall asleep, the review begins. What seemed forgotten during the day may simply have been waiting for the quiet. The word plasticity in brain science does not mean fragility. It means capacity for change. Neuroplasticity refers to the brain's lifelong ability to form new connections between neurons, strengthen existing ones, and prune pathways that are no longer used. For most of human history, scientists believed the adult brain was fixed, that the neurons you had at age 20 were the ones you kept, declining steadily from there. That understanding changed in the 1960s and accelerated through the 1990s, supported by a growing body of cellular and imaging research. The brain, it turns out, is one of the most adaptive structures in the known natural world. Sleep is not a pause in neuroplasticity. It is its most productive phase. During waking hours, the brain is in acquisition mode, absorbing information, reacting to stimuli, forming initial connections. During sleep, particularly in the deeper stages, it shifts into consolidation mode, refining, strengthening, and integrating the day's learning into longer-term architecture. Studies in human participants have shown that sleep after learning improves retention significantly compared to equivalent rest periods while awake. The brain does not simply store what it learned. It builds on it, reconstructs it, and deepens it in the dark. There are 2 primary types of sleep, and both contribute to plasticity in different ways. Non-REM sleep, which includes the deepest stages of slow-wave sleep, handles structural consolidation, moving information from short-term hippocampal storage toward more stable cortical networks. REM sleep, which tends to dominate in the later hours of the night, handles integration, weaving new learning into existing frameworks, finding patterns, building connections between disparate memories. A full night of sleep cycles through both multiple times. Shortening sleep by even 90 minutes can significantly reduce the amount of REM the brain receives. affecting both emotional memory and creative integration. The hippocampus is the brain's short-term memory hub, shaped roughly like a seahorse and located deep in the temporal lobe. During the day, it captures incoming information in a fast, flexible encoding system, like a scratch pad that receives everything first. During slow-wave sleep, the hippocampus replays this information to the cortex in a compressed, rapid sequence. The cortex gradually absorbs and integrates this content into its slower, more durable storage system. Think of the hippocampus as the night shift worker, transferring files from a temporary drive to the permanent archive while the rest of the system rests. Sharp wave ripples are one of the clearest signatures of memory consolidation in the sleeping brain. These are brief, high-frequency bursts of neural activity, lasting about 100 milliseconds, generated in the hippocampus during slow-wave sleep. When researchers record neural activity during sleep and identify these ripples, they find that the neurons firing during a ripple are the same neurons that fired during a specific experience earlier in the day. The brain is quite literally replaying what it lived. Each ripple is a compressed rerun of something real. A conversation, a route, a problem, stripped of its context and passed forward. Sleep spindles are a hallmark of the consolidating brain. Brief bursts of synchronized neural activity in the 12 to 15 Hz range that appear during stage 2 non-REM sleep. They were identified in the 1930s, but their function was not fully understood until much later. Research now suggests that spindles protect the brain's consolidation work from being disrupted. They act as a kind of neural firewall, temporarily reducing the brain's responsiveness to external stimuli so the internal transfer of memories can proceed undisturbed. Individuals who produce more spindles per hour of sleep consistently show better memory performance the following day. The glymphatic system is one of the most consequential discoveries in neuroscience in the past 20 years. Announced in research from the University of Rochester in 2013, this system functions as the brain's waste clearance network, a series of fluid channels running alongside blood vessels that flush cerebrospinal fluid through brain tissue, carrying out metabolic waste products. The glymphatic system appears most active during sleep, particularly during slow-wave sleep, when brain cells may shrink slightly, expanding the spaces between them and allowing fluid to flow more freely. The full significance of this system for long-term neurological health is still being investigated. Among the waste products studied in relation to the glymphatic system is beta amyloid, a protein fragment that accumulates in the brain over time and has been associated with neurodegenerative processes in animal models and some human studies. The relationship between sleep, glymphatic clearance, and long-term brain health is an active and evolving area of research. The precise mechanisms in humans are not yet fully established, and researchers continue to investigate the degree to which sleep-dependent clearance contributes to healthy aging. What the evidence does suggest is that the brain's maintenance process is not passive, and that sleep appears to play a meaningful role in it. So yes, the brain rewiring at 3 in the morning is not metaphor, it is mechanism. By this point in tonight's journey, the picture is already emerging. Sleep is the period when the brain runs its most complex maintenance operations simultaneously, consolidating memory, refining connections, and clearing metabolic byproducts through a fluid system that only fully opens in deep sleep. What you have always called rest is, underneath, coordinated biological work. The fear of decline may be partly a fear that this work is no longer happening. Tonight, the system running beneath your closed eyes is evidence that it is. Long-term potentiation, often abbreviated LTP, is the cellular mechanism underlying much of what we call learning and memory. When two neurons fire together repeatedly, the connection between them strengthens. The synapse becomes more sensitive, more efficient at transmitting signals. This is what the phrase neurons that fire together, wire together refers to. Sleep amplifies this process. During slow-wave sleep, the brain selectively strengthens the synaptic connections formed during the day's learning while allowing weaker, less reinforced connections to fade, a process researchers call synaptic homeostasis, which keeps the system balanced and capable of continued growth. Synaptic homeostasis theory, proposed by researchers Giulio Tononi and Chiara Cirelli, suggests that waking life gradually strengthens synapses throughout the brain. Learning accumulates as connection strength increases, and that sleep serves partly to reset this overall level by downscaling connections globally. This prevents the system from becoming saturated. Imagine a classroom where every student keeps raising their hand at once. Eventually, no one can be heard. Sleep quiets the room selectively, so the most relevant connections stand out more clearly in the morning. The theory remains an area of active scientific discussion, with ongoing research refining its scope and predictions. The prefrontal cortex, the region associated with judgment, planning, and working memory, is one of the most sleep-sensitive areas of the brain. Even one night of poor sleep measurably affects prefrontal function, reducing performance on tasks requiring sustained attention, flexible thinking, and emotional regulation. This is not simply tiredness. It reflects a genuine reduction in the cortex's operational capacity. The prefrontal cortex also takes longer than many other regions to come fully online in the morning. The slow, foggy feeling after poor sleep is partly this region waiting to reach its normal working level of function. REM sleep, rapid eye movement sleep, gets its name from the visible movement of the eyes beneath closed lids during this stage. First formally identified in the 1950s by researchers Nathaniel Kleitman and Eugene Azarinsky, REM sleep is associated with vivid dreaming, emotional memory processing, and the creative integration of new information with existing knowledge. The brain during REM shows activity patterns that closely resemble waking states in some respect, yet the body is largely paralyzed. Atonia, preventing physical movement in response to dream content. REM periods lengthen over the course of the night, meaning the final hours of sleep contain disproportionately more of it. The default mode network is a set of interconnected brain regions that become most active when the brain is not focused on an external task. During daydreaming, self-reflection, imagining the future, and during certain phases of sleep, this network plays a role in integrating personal narrative, connecting new experiences to the existing story of the self. Research has found that REM sleep activates the default mode network in ways that may support the emotional processing of difficult experiences. weaving them into a broader autobiographical context rather than leaving them as isolated, unintegrated events. It is, in some sense, the brain rereading its own story overnight. The amygdala, the brain region most closely associated with emotional memory and threat detection, shows reduced reactivity after adequate sleep compared to sleep-deprived states. Studies using functional imaging have found that the amygdala responds more strongly to negative emotional stimuli after sleep deprivation, and that the prefrontal cortex's regulatory influence over it weakens. In other words, the brake on emotional reactivity partly depends on sleep. The irritability, low tolerance, and emotional fragility that accompany poor sleep are not simply subjective feelings. They reflect measurable changes in how the brain processes and regulates its own emotional responses. Brain-derived neurotrophic factor, often written BDNF, is a protein that supports the survival and growth of neurons and plays a central role in the biological process of neuroplasticity. It is sometimes described as fertilizer for the brain. Sleep appears to influence BDNF levels. Research in animal models has found that sleep deprivation reduces BDNF expression in areas associated with memory and learning, while certain stages of sleep have been associated with increased production. The relationship continues to be clarified in human research, but BDNF sits at the intersection of sleep, plasticity, and the brain's long-term capacity to maintain itself. Cortisol, the hormone associated with the stress response, follows a distinct daily cycle that intersects with sleep architecture in ways that matter for brain function. Cortisol levels are typically at their lowest in the first hours of sleep and rise toward their peak in the early morning hours, reaching maximum concentration around the time of waking. This rise contributes to alertness and the return of focused cognition. Chronically elevated cortisol, from sustained psychological stress, can interfere with both sleep quality and hippocampal health over time. The brain's plasticity is not isolated from the body's stress systems. They are deeply interwoven. Norepinephrine is a neurotransmitter that plays a role in arousal, attention, and the consolidation of emotional memories. During waking hours, norepinephrine is present throughout the brain, contributing to vigilance and stress responses. During REM sleep, something unusual happens. Norepinephrine levels drop to their lowest point in the entire sleep-wake cycle, near zero in some brain regions. This chemical quiet during REM may be important for the processing of emotional memories. Some researchers have proposed that it allows the brain to reactivate and reintegrate emotional experiences without the distress signal that norepinephrine carries, effectively re-encoding the memory with reduced emotional charge. The hippocampus does not simply replay recent memories during sleep. It appears to replay them selectively, prioritizing the most emotionally significant or reward-associated experiences. Research in animals has found that neurons representing an interesting or novel location fire more frequently during subsequent sleep than neurons representing an unremarkable one. The brain is not an unbiased archivist. It edits as it stores, giving additional processing time to what mattered most. The memories that feel most persistent in the morning may partly reflect the quiet editorial decisions the sleeping hippocampus made during the night. Place cells are neurons in the hippocampus that fire when an animal occupies a specific location in space. Essentially, a neural GPS system. In landmark research by John O'Keefe and colleagues, and later extended by Matt Wilson and Bruce McNaughton, scientists found that place cells active during a rat's daytime exploration fired in the same sequence during subsequent sleep, but compressed at roughly 10 to 20 times the waking speed. This was among the first direct evidence that the sleeping brain replays waking experience. It opened a new chapter in the science of memory, and O'Keefe's work eventually contributed to a Nobel Prize in Physiology or Medicine in 2014. Motor learning, the acquisition of physical skills, also consolidates during sleep in ways that mirror declarative memory. People trained on a finger-tapping sequence in the morning and tested 12 hours later showed minimal improvement if kept awake, but significant improvement after a night of sleep. The same task trained in the evening and tested after sleep showed even larger gains. The improvement was not due simply to practice. It occurred during the offline period of sleep. Motor cortex activity during post-training sleep appears to replay and refine the movement patterns, strengthening the neural circuits that will execute them more efficiently the next day. Language acquisition in children shows particularly clear dependence on sleep for consolidation. Young children who napped after learning new words retained them significantly better than those who stayed awake for an equivalent period. Infant and toddler brains show proportionally more slow-wave and REM sleep than adult brains, a pattern consistent with the enormous demands placed on the developing brain by early learning. Sleep is not a break from learning in childhood. It is the medium through which the most fundamental learning becomes permanent. The hours of quiet in which the brain sorts, stores, and builds the architecture of language. The aging brain and sleep are connected in ways that have been studied with increasing precision. As people age, the proportion of slow-wave sleep typically decreases. There is less of the deepest, most restorative non-REM sleep. Some research suggests this reduction may be associated with changes in memory consolidation efficiency, though the relationship is complex and individual variation is substantial. Critically, the capacity for neuroplasticity does not vanish with age. The brain retains the ability to form new connections throughout life. What changes over time is the efficiency of certain maintenance processes, not the fundamental underlying capacity for adaptation. Across the first 25 facts, a coherent picture has formed. The sleeping brain is not a passive system awaiting morning. It is actively consolidating memory through hippocampal replay and sharp wave ripples, refining synaptic connections through potentiation and homeostasis, clearing metabolic waste through the glymphatic system, and processing emotional experience through the norepinephrine quiet of REM. The fear of memory decline and brain aging, the fear that brought many listeners here tonight, is partly a fear that this maintenance work is failing. What the science reveals is that sleep is the single most available lever for supporting it. The work is still happening. You are evidence of that right now. You've been with us for a while now, and your breathing has likely softened without you noticing. A soft subscribe helps another restless mind find this sanctuary too. Now, let's pause together, one sleepy soul at a time. Right now, the hippocampus in your brain has been doing exactly what we described, running its quiet review of the day. firing in soft sequences, passing forward what it decides to keep. You don't need to direct it. It already knows what it is doing. The question that just opened, about what happens when that review meets years of interrupted sleep, of nights that weren't long enough, lives inside this chemistry. Inside what accumulates when the maintenance window keeps getting shortened. Notice your breath. Notice that the system continues its work regardless. Even now. Even here. Now we continue. Sleep does more than consolidate what was learned. It can solve problems that remained unsolved before sleep. Research by Ulrich Wagner and colleagues found that participants who slept between two problem-solving sessions were significantly more likely to discover a hidden shortcut in a mathematical task than those who remained awake for an equivalent period. The brain, during sleep, appears to reorganize information in ways that reveal underlying structure not apparent during focused waking thought. The phrase "sleep on it" describes something real. The sleeping brain is not merely resting from the problem. It is, in a measurable sense, working on it. The term offline processing is used in memory research to describe the brain's consolidation work during sleep, in contrast to online processing, which happens during active waking engagement. During offline processing, the brain is free from the demands of incoming sensory information. It can run operations that would be interrupted or distorted by real-time experience. Some researchers compare it to a computer running maintenance processes overnight, defragmenting, backing up, updating, in a window when the system is not needed for other tasks. The analogy has limits, but the basic logic holds. Targeted memory reactivation is a technique that researchers have used to investigate the specificity of sleep-based consolidation. In a typical experiment, participants learn information while a specific odor or sound is present in the background. During subsequent slow-wave sleep, the same cue is introduced at low intensity, below the threshold of waking. Participants who receive the cue during sleep show improved memory for the information associated with it the following morning, compared to those who do not. The sleeping brain responds to environmental cues and uses them to selectively amplify specific memory traces. The two-stage model of memory consolidation, proposed in various forms over the past 3 decades, suggests that memories are first encoded in the hippocampus and later transferred to the neocortex for long-term storage. Sleep, specifically the interplay between slow-wave sleep and REM sleep, facilitates this transfer. The model helps explain why memories initially feel fragile and context-dependent, and why they become more stable, flexible, and integrated over time. It also explains why disrupting sleep in the days following a learning event can impair retention even if the initial encoding seemed intact. The cerebellum, often associated with movement coordination, also plays a role in the consolidation of procedural and motor memories during sleep. Research has found that cerebellar activity during post-training sleep correlates with subsequent improvement in skilled movement tasks. The cerebellum's contribution to sleep-dependent consolidation has historically received less attention than the hippocampus, but its role is increasingly recognized as significant, particularly for physical skills, timing, and the fine-grained sequence learning that underlies everything from playing an instrument to handwriting. The sleeping brain refines coordination as thoroughly as it refines memory. REM sleep has been specifically associated with the consolidation of emotionally charged memories and with the updating of threat response patterns. Research suggests that the amygdala and hippocampus communicate extensively during REM, processing the emotional dimensions of experience in a context where the stress hormone norepinephrine is suppressed. This may explain why sleep deprivation is associated with a tendency to overestimate threat and underestimate safety. The REM-dependent recalibration of emotional memory has not occurred, and the system remains tuned, still responding to the previous evening's most recent alarm. Slow oscillations are large. Rhythmic fluctuations in electrical activity across the cortex during deep, slow-wave sleep, alternating between periods of high neural activity and near silence, cycling roughly once per second. These oscillations appear to coordinate the timing of hippocampal sharp-wave ripples and thalamo-cortical sleep spindles, creating a synchronized window for memory transfer. The slow oscillation acts as a kind of conductor, ensuring that the different instruments of consolidation fire at the right moments in the right sequence. Disrupting these oscillations, which can be done through gentle electrical stimulation during sleep, selectively impairs memory consolidation. The thalamus, a deep brain structure that functions as the primary relay station for sensory information, plays an active role in sleep. During sleep, the thalamus generates the rhythmic bursts of activity associated with sleep spindles, and it coordinates the broader gating of sensory input that makes deep sleep possible. The thalamus essentially acts as a sensory filter, reducing the brain's responsiveness to external stimulation so that internal consolidation work can proceed without interruption. This is one reason why sleeping in a noisy or unpredictable environment degrades sleep quality, even when the person does not wake fully. The thalamus is repeatedly pulled toward external monitoring. Sleep architecture. The pattern and timing of sleep stages across the night is not uniform. A typical night moves through several 90-minute cycles, each containing a sequence of non-REM stages followed by a period of REM. In the first half of the night, slow-wave sleep dominates. In the second half, REM periods lengthen and slow-wave sleep decreases. This architecture means that different memory systems receive their most intensive processing at different points in the night, cutting sleep short, even by the final 90 minutes. Disproportionately removes the REM-heavy second half, affecting the integration and emotional processing functions it supports. Procedural memory, the kind underlying skills and habits, is handled differently from declarative memory during sleep consolidation. While declarative memories rely heavily on hippocampal neocortical transfer during slow-wave sleep, procedural memories are thought to involve striatal and cerebellar circuits more prominently. This distinction has practical implications. The skill practiced before bed benefits from a different overnight process than the facts studied before bed. Both processes run simultaneously during a full night of sleep, which is one reason that 7 to 9 hours supports a broader range of cognitive function than any shorter window. The concept of sleep-dependent learning has expanded significantly, with research showing that even brief naps can support memory consolidation. A 90-minute nap taken in the afternoon that includes both slow-wave and REM sleep can produce consolidation benefits comparable to a full night in some tasks. A shorter nap of 60 minutes containing slow-wave sleep alone can benefit declarative memory. These findings suggest that the consolidation mechanisms activate whenever the appropriate sleep stages are reached, not exclusively during nocturnal sleep. The brain does not insist on night. It uses whatever sleep is available. Neurogenesis, the birth of new neurons, occurs in the adult brain primarily in two regions: the olfactory bulb and the hippocampus. In the hippocampal dentate gyrus, new neurons are continuously generated and, if they survive and integrate into existing circuits, contribute to memory encoding and pattern separation. Sleep appears to support the survival and integration of newly born neurons. While chronic sleep deprivation in animal models has been associated with reduced rates of hippocampal neurogenesis, the picture in humans is less clear and continues to be studied. Adult human hippocampal neurogenesis is real but more limited than in rodents, and its functional significance is debated. The connection between sleep and creativity has been documented in experiments and reported by some of the most productive minds in scientific and artistic history. The chemist Auguste Kekulé reportedly described understanding the ring structure of benzene through a dream image. Paul McCartney has said the melody for Yesterday came to him in a dream. The conditions of REM sleep: reduced norepinephrine, active default mode network, weakened prefrontal inhibition, may create an environment where unusual associations between stored memories are more easily formed. What the waking mind might reject as illogical, the sleeping mind may follow to a genuinely useful connection. Prospective memory, the ability to remember to do something in the future, is also strengthened by sleep. Experiments have shown that people who sleep after forming an intention are better at executing it later than those who stayed awake for an equivalent period. The sleeping brain appears to tag future-oriented memories with a kind of forward salience, making them more accessible when the relevant context arises. This is one of the more practically significant findings in sleep research. Touching on everyday experience. The plan that felt clear at bedtime that somehow surfaces unbidden at exactly the right moment. Looking across facts 26 to 40, the evidence for sleep's role in memory, plasticity, and cognitive function has only grown stronger over the past 3 decades. What was once considered a passive biological necessity has been revealed as an active, coordinated, multi-system process. Problem solving, creative integration, emotional recalibration, procedural refinement, and prospective memory enhancement, all occurring in the same 90-minute cycles, night after night. The brain's resilience is not simply a property of its waking hours. It is built, maintained, and quietly renewed in the dark. There's a quietness settling in now, the kind that comes when facts have been absorbed rather than held. Take a slow breath. Notice how the body has softened without any instruction. We're traveling through the science of the sleeping brain together, you and I, in the very state the science describes. Let's continue. The sleeping brain does not simply consolidate what it learned during the day. It also forgets. And forgetting is not a failure of the system. Intentional forgetting, or memory pruning, removes connections that are redundant, outdated, or irrelevant, keeping the system efficient and reducing interference between similar memories. During sleep, weakly encoded or infrequently accessed traces are allowed to decay, while strongly encoded ones are reinforced. The brain that wakes tomorrow is not a complete record of everything that entered it. It is an edited, curated version, shaped by the priorities the sleeping consolidation process assigned. Schema: the organized frameworks of prior knowledge through which new information is interpreted, interact with sleep consolidation in a specific way. When new information fits an existing schema, it consolidates faster and integrates more deeply. When it contradicts a schema, it may require more sleep cycles to be fully absorbed, and it is more likely to prompt a dream that attempts to reconcile the contradiction. Learning that fits what you already know sleeps well. Learning that challenges what you believe takes longer to settle. The resistance is not stubbornness. It is the brain protecting the architecture it has already built. The sleeping brain processes language differently from the waking brain. Studies using neuroimaging have found that meaningful words presented during sleep at low volumes can activate language processing regions, while nonsense words do not produce the same response. The sleeping brain retains a degree of semantic processing capacity. It distinguishes meaning from non-meaning, even in the absence of conscious awareness. This finding has implications for understanding what kind of auditory input during sleep might facilitate or interfere with consolidation, and why sleep environments with meaningful speech, television, conversation tend to disrupt rest more than ambient sound. Theta waves, neural oscillations in the 4 to 8 Hz range, are prominent in the hippocampus during active spatial navigation and memory encoding while awake. They also appear during REM sleep, particularly in the hippocampus and adjacent structures, where they may coordinate the replay and integration processes occurring during that stage. The reappearance of theta during REM has led some researchers to propose that REM sleep is functionally similar to an exploratory waking state, the brain navigating and integrating its internal map of experience rather than the external map of the world. Deep sleep has been shown in multiple studies to improve performance on standardized tests when it follows learning. Students who slept between study sessions showed better retention than those who spent the same time reviewing material while awake. This has practical significance beyond academic performance. The implication is that the common strategy of reducing sleep to create more study time actively undermines the consolidation of what was studied. The hours of sleep are not neutral. They are, for much of what is learned, the most productive hours of the learning process. Acetylcholine, a neurotransmitter associated with arousal, attention, and memory encoding, oscillates dramatically across the sleep-wake cycle in ways that directly shape consolidation. During waking and REM sleep, acetylcholine levels are high, facilitating encoding and integration. During slow-wave sleep, they drop sharply. This drop is thought to be a necessary condition for hippocampal to cortical transfer. High acetylcholine keeps information circulating within the hippocampus, while low acetylcholine allows it to flow outward to the cortex. The chemistry of deep sleep is not simply suppression. It is a specific electrochemical state tuned for memory transfer. The pineal gland produces melatonin, the hormone that signals darkness and promotes the onset of sleep. But melatonin does not cause sleep directly. It lowers core body temperature and shifts the brain toward a state conducive to sleep onset. The temperature drop associated with melatonin release is one of the reasons that a cool bedroom environment facilitates better sleep. It aligns with the body's own thermoregulatory signal. Conversely, exposure to blue wavelength light in the evening suppresses melatonin release, delaying sleep onset and shifting the timing of the night's consolidation work by the same margin. Adenosine is a chemical that accumulates in the brain throughout waking hours as a byproduct of neural activity, often described as sleep pressure. The longer you are awake, the higher the adenosine concentration, and the stronger the drive to sleep. Caffeine works by blocking adenosine receptors, preventing the signal from reaching its destination. When caffeine's effect wears off, the accumulated adenosine floods those receptors simultaneously. The crash. Sleep clears adenosine from the brain, resetting the pressure to near zero. One of the functions of the glymphatic system may be to facilitate this clearance. Though the precise mechanisms continue to be studied, the suprachiasmatic nucleus, a small cluster of neurons in the hypothalamus, just above the point where the optic nerves cross, is the brain's master clock. It receives light information from the eyes and uses it to synchronize the body's circadian rhythms to the external day-night cycle, disrupting this synchronization through shift work, frequent travel across time zones, or irregular sleep schedules does not simply cause tiredness. It misaligns the timing of nearly every biological process in the body, including the hormonal and neurochemical conditions that support consolidation. The brain's maintenance window depends on the clock running at the right time. Halfway through this journey, the answer is sharpening. The brain that rewires at 3 in the morning does so because 3 in the morning is precisely when the conditions converge. Norepinephrine at its lowest, acetylcholine quieted, slow oscillations coordinating hippocampal ripples and cortical spindles, glymphatic channels wide. The fear of decline is real. But what the science shows is that the brain has built an extraordinarily elaborate response to that challenge, running every night in the right conditions, with more sophistication than any intervention yet devised. The question the second half of tonight follows is, what does the system do when it is given what it needs? What just opened? The question of what happens when the brain's maintenance window runs undisturbed, night after night, through the full depth of slow wave and into the long REM of early morning, is not only a neuroscience question. You may be carrying a version of the answer right now. Somewhere between the worry that things are slipping and the body that is, even now, beginning its quiet work. That distance between the fear and the fact is what the second half of tonight moves through. Notice your breath. Feel that the inhale comes without instruction. Feel that the system is already demonstrating its own resilience. Now we continue. The brain rewires most efficiently when sleep follows learning within a few hours. Research has found a window in which consolidation begins, typically within the first sleep cycle after acquisition. Information encoded in the morning that reaches sleep by nightfall consolidates more completely than information encoded in the evening that waits until the following night. The brain does not store experiences indefinitely in their original form, waiting for sleep. They begin to degrade in the hippocampus almost immediately. Sleep interrupts that degradation and transfers what remains into more stable architecture. Emotional regulation and sleep form a feedback loop that runs in both directions. Good sleep supports emotional regulation. Effective emotional regulation, in turn, reduces the nighttime arousal that interrupts sleep. When this loop is disrupted, as it often is by anxiety, unresolved stress, or hypervigilance, the system struggles to establish the deep sleep stages that process emotional memory. The result can be a self-reinforcing pattern. Poor sleep increases emotional reactivity, which increases difficulty sleeping, which increases emotional reactivity. Understanding the loop is part of interrupting it. The prefrontal cortex, which provides inhibitory control over the amygdala, is restored by sleep in ways that directly affect the ratio of threat detection to threat assessment. A well-rested prefrontal cortex can evaluate a perceived threat, contextualize it, and modulate the response. A sleep-deprived prefrontal cortex passes the signal through with less filtering. This is why decisions made in a state of sleep deprivation tend toward risk aversion or impulsivity, two sides of the same diminished cortical regulation. The brain that wakes after deep sleep is genuinely better at being a brain. The brain's default mode network, which processes self-referential thought and autobiographical memory, is active during certain phases of sleep in ways that may support what some researchers call narrative consolidation, the integration of new experience into the ongoing story of the self. Dreams, particularly REM dreams, often feature the dreamer navigating emotionally significant scenarios drawn from recent and distant memory. The content is not random. It reflects the active work of contextualizing new events within accumulated personal history, finding where the new experience belongs in the architecture of what came before. Music and rhythm may influence the brain's consolidation processes through a phenomenon called neural entrainment, the tendency of neural oscillations to synchronize with external rhythmic input. Some researchers have explored whether slow rhythmic auditory stimuli during slow-wave sleep can amplify the brain's own slow oscillations, potentially enhancing consolidation. Results have been mixed, and the field is still developing. But the underlying mechanism, that the sleeping brain remains responsive to rhythmic sensory input, and that this responsiveness can influence internal oscillatory patterns, is well established. The relationship between exercise and sleep-dependent plasticity is bidirectional. Aerobic exercise increases BDNF levels, supports hippocampal health, and improves sleep quality and architecture in multiple studies. Sleep, in turn, supports the physical recovery and motor consolidation that make exercise beneficial. The two systems appear to amplify each other. Adequate sleep makes the brain more responsive to the plasticity-promoting effects of exercise, and regular exercise deepens the sleep stages most associated with consolidation. Neither works at its full capacity without the other. Stress hormones and sleep architecture interact in ways that can create or dissolve cognitive reserve over time. Chronic stress elevates cortisol, which suppresses slow-wave sleep, which reduces glymphatic activity, which allows waste products to accumulate that would otherwise be cleared. The pathway from sustained stress to changes in cognitive function runs partly through the degradation of sleep quality. This is not deterministic. Many people maintain healthy sleep, even under high stress, through behavioral and environmental supports. But the mechanism is real, and the cumulative effects of repeated disruption are measurable in long-term studies. The sleeping brain's responsiveness to emotional context persists into deep sleep. Research has found that people presented with emotionally charged words during slow-wave sleep show measurable neural responses, activation in the amygdala and related structures, even in the absence of conscious awareness. Neutral words do not produce the same response. The brain, even at its least conscious, continues to monitor its environment for signals of potential emotional significance. This is one reason why sleep environments with conflict, tension, or emotional unpredictability can alter sleep architecture, even when the sleeper does not consciously register the disturbance. The role of sleep in immune function and brain health intersects in ways that have become more visible in recent research. Inflammatory cytokines, signaling molecules released during immune responses, can disrupt sleep architecture, and disrupted sleep can, in turn, affect inflammatory signaling. The brain and immune system communicate through pathways that run through the same blood-brain barrier that the glymphatic system helps to maintain. Sleep deprivation has been associated in some studies with increased markers of neuroinflammation. Though the direction of causality is complex and the mechanisms continue to be studied, the sleeping brain is not silent in terms of sensory processing. It continues to evaluate certain inputs and can respond to them selectively. A sleeping parent wakes more readily to the sound of their child's voice than to a sound of equal volume that lacks personal significance. The brain's monitoring during sleep is not random vigilance. It is tuned to what the waking brain has identified as most worth protecting. This selective responsiveness exists on a continuum from full wakefulness to deep sleep, with the threshold for response rising as sleep deepens and the selectivity increasing at the same time. Memory reconsolidation is distinct from initial consolidation. When a stored memory is retrieved, brought back into active awareness, it becomes temporarily labile again, requiring another round of consolidation to restabilize. Sleep after retrieval may affect the quality of this reconsolidation, potentially altering the memory's subsequent content, emotional tone, or accessibility. This process has implications for understanding how memories change over time. Why a memory recalled frequently across years may gradually shift in detail or emotional weight, while a rarely recalled memory may remain more stable in its original form. Dreaming appears to serve some of the same integration functions as waking cognition, but with the constraints of logical consistency and goal-directedness loosened. This loosening may be adaptive. By following associations that waking thought would reject as impractical, the dreaming brain explores a larger space of possible connections between stored memories. Some of these connections are irrelevant. Some may be useful in ways that are not immediately apparent. The experience of waking from a dream with an unexpected solution to a problem that resisted waking effort is not merely anecdotal. It reflects a real feature of REM state processing. The aging brain retains plasticity, but the conditions required to express that plasticity change. Older adults often experience reduced slow-wave sleep, lighter and more fragmented sleep architecture, and earlier morning awakening. These changes can reduce the efficiency of hippocampal cortical transfer and glymphatic clearance. but the mechanisms remain available. Research has found that older adults who maintain sleep quality through behavioral sleep hygiene, management of sleep disorders, and reduction of fragmentation show cognitive trajectories more similar to those of younger good sleepers than to age-matched poor sleepers. The brain's capacity to maintain and renew itself does not vanish. The conditions for it require more deliberate support. Individual variation in sleep architecture and consolidation efficiency is substantial. Some people show robust memory consolidation after relatively modest amounts of slow-wave sleep. Others require more. Some people's brains generate more sleep spindles per hour of sleep than others, and higher spindle density has been associated with better consolidation performance. These variations are partly genetic. Studies of twins have found significant heritability in sleep architecture measures, but they are also shaped by age, health, and the cumulative history of sleep behavior. There is no single profile of optimal sleep. There is only the individual brain and the conditions it has been given. We now understand that the brain rewiring at 3 a.m. is not a single process. It is a coordinated ensemble. Slow oscillations orchestrating the transfer, spindles protecting the signal, ripples delivering the specific memory trace, glymphatic channels clearing the residue, REM weaving the emotional dimensions into a larger narrative. The fear of memory decline is partly a fear that this ensemble has stopped playing. What the research shows is that it continues to play throughout life, with changes in tempo, changes in instrumentation, but the same underlying music. Tonight, it is playing. Sleep timing, the alignment of sleep with the circadian rhythm, may matter as much as sleep duration for consolidation efficiency. Sleeping at a time misaligned with the body's internal clock, even for the same number of hours, produces sleep of lower quality and reduced consolidation benefit. The brain's maintenance systems are calibrated to run at specific circadian phases. The deep, slow-wave stages are most reliably reached in the early part of the night sleep window, and the long REM stages in the second half. Shifting sleep by several hours disrupts both. The clock and the calendar cooperate. The sleeping brain runs what some researchers describe as a global reset each night, not of the memories themselves, but of the brain's sensitivity to new learning. By the time a person wakes after adequate sleep, synaptic scaling has reduced the overall weight of the day's newly formed connections, freeing the system to encode the next day's experience without interference from the previous day's traces. This homeostatic function of sleep, resetting the learning surface, may be as important as the consolidation function. Without it, the brain would accumulate saturation, becoming progressively less able to form new memories. Sound sleep environments support the brain's maintenance work not only by reducing external disruption, but by creating the internal conditions for deep sleep onset. Temperature, light, noise, and psychological safety all contribute to the speed with which the brain moves from waking into the slow-wave stages where consolidation begins. A bedroom that feels safe, that the sleeping brain has learned to associate with rest rather than vigilance, reaches deep sleep faster and maintains it longer. The brain is watching the environment even as it sleeps, and an environment that requires watching prevents the deepest stages of sleep from fully developing. The phrase "sleeping on a problem" describes a real and measurable phenomenon in cognitive research, but it has a specific architecture. The enhancement in problem solving that follows sleep is most associated with REM sleep, not simply rest. The creative recombination of information that occurs during REM, in the context of reduced prefrontal inhibition and active default mode network, generates novel associations that structured waking thought may not produce. The benefit is not passive. It requires the brain to have engaged with the problem before sleep, giving the consolidating system something to work with during the night. The brainstem structures that generate the basic rhythms of sleep, the pons, the locus coeruleus, the raphe nuclei, are among the oldest parts of the brain in evolutionary terms. The consolidation machinery they support predates the development of the neocortex by hundreds of millions of years. Sleep appears to be one of the most deeply conserved biological behaviors in animal life, present in some form in every studied vertebrate and many invertebrates. The fact that such an ancient system has been preserved across such divergent evolutionary paths suggests that it provides something irreplaceable, something no waking process has managed to replace. Memory is not stored in a single location. A complex memory, a meal with someone important, a moment of unexpected kindness, involves sensory details processed by the sensory cortices, emotional dimensions processed by the amygdala, spatial and temporal context encoded by the hippocampus, and meaning embedded in the semantic networks of the left temporal lobe. When the hippocampus replays this memory during sleep, it reactivates all of these components simultaneously, and the cortical regions receiving the signal update their representations to reflect the new context. Memory is not a file. It is a distributed pattern that sleep gradually consolidates into coherence. The sleeping brain's experience of time is compressed. What the hippocampus replays in a sharp wave ripple, lasting a few hundred milliseconds, may represent an experience that unfolded over minutes. What a dream compresses into what feels like an extended narrative may represent only seconds or minutes of actual sleep time. The brain does not replay experience at real-time speed during consolidation. It extracts the structure, the sequence, the emotional weight, and passes those forward. The full sensory texture of the original experience is not preserved. What remains is the meaning and the shape. The connection between sleep and chronic pain has become an active research area. Poor sleep lowers pain thresholds and reduces the brain's ability to modulate pain signals through descending inhibitory pathways. Chronic pain disrupts sleep, and disrupted sleep amplifies pain. Another bidirectional loop with potential for reinforcement. The brain regions involved in both processes, the anterior cingulate cortex, the thalamus, the prefrontal cortex, are the same regions most affected by sleep loss. Addressing sleep quality has become an explicit component of some chronic pain management frameworks, recognized as a primary intervention target rather than a secondary concern. The sleeping brain's maintenance work includes more than memory and emotion. It repairs myelin, the insulating sheath around neurons that allows signals to travel efficiently. Through the activity of oligodendrocyte precursor cells that divide preferentially during sleep in animal models, it regulates the expression of genes involved in synaptic function. immune response, and cellular repair. It restores the brain's supply of glycogen, the local energy reserve for neurons. Sleep is not a single function. It is the umbrella under which an enormous number of restoration processes run concurrently, all of them requiring the same basic condition: the brain not being needed for anything else. The brain that rewires at 3 in the morning has taught us something that shifts how we understand the relationship between rest and productivity, between sleep and cognition, between the night and everything that happens after it. Sleep is not the absence of wakefulness. It is its own kind of work, precise, necessary, irreplaceable. The fear of memory decline, of aging, of losing what makes you yourself. That fear has a partial answer in the science of sleep. Not a cure, not a guarantee, but a practice, giving the system the conditions it needs to do what it has been doing every night. You might be feeling tired now. That's perfectly fine. Breathe slowly. Feel the cycles. The hippocampus replays the day. The cortex receives and absorbs. The glymphatic channels open and clear. All of it moving. All of it quiet. All of it cycling through the same deep rhythm, night after night. Like your breath. Inhale. Exhale. Inhale. Exhale. A small cycle within the vast repair. You are part of that movement. You don't need to hold on to anything. Let everything continue in its own order, like the slow oscillation moving through the sleeping cortex. Unhurried, unresisting, simply existing. We're almost there now. Every morning you wake is partly a product of the night before. Not just in terms of rest, in terms of what was consolidated, what was cleared, what was refined while you were elsewhere. The person who opens their eyes has been quietly assembled in the dark. The brain worked while you were gone, and what it built is what you wake into. The hippocampus is small, roughly the size of a finger. The amount of work it does in a single night of sleep is disproportionate to its size. It replays. It compresses. It transfers. It holds the day's experience long enough for the cortex to absorb it, then releases what it held. Every morning, it begins again. Sharp wave ripples last less than a second each. In a night of healthy, slow-wave sleep, tens of thousands of them occur. Each one carries a trace of something real, something lived, something seen, something felt. The sleeping brain is not idle. It is working through what the day left behind, one ripple at a time. Memory is not the past. It is a reconstruction that happens in the present, using traces that sleep has helped to stabilize. When you remember something clearly and warmly, you are partly remembering the sleep that followed it, the night that held what the day gave, and passed it forward. The glymphatic channels open wider in sleep than at any other time. The brain that relaxes deeply enough to enter slow-wave sleep gives its own clearance system access it otherwise cannot have. What accumulates during the day begins to move. Rest is not avoidance. It is the opening of a door that stays closed while you are awake. Neuroplasticity does not require youth. It requires the conditions that allow the brain to form new connections, stabilize them, and integrate them into existing architecture. sleep is the most foundational of those conditions. It does not disappear with age, but it does require more deliberate protection as the years continue. The norepinephrine that carries alarm through the brain during the day reaches its lowest point during REM sleep. In that quiet, the brain revisits what alarmed it, not to repeat the alarm, but to reduce it. The emotional memory returns, but in a different chemical environment. REM sleep is not the continuation of the day's anxiety. It is, slowly and quietly, its partial resolution. Sleep spindles protect consolidation the way silence protects thought. Without the brief suppression of external signals they provide, the brain's internal transfer work would be interrupted before it could complete. The body's stillness during deep sleep is not incidental. It is part of the mechanism. The stillness and the work are the same thing. BDNF, the protein sometimes called fertilizer for the brain, is produced and released during sleep. According to research in animal models, the brain that rests may be growing the support structures for its own neurons. Rest is not the opposite of growth. In the brain, rest is often where the conditions for growth are quietly assembled. The slow oscillation of deep sleep moves through the cortex like a wave. A second of activity, a second of near silence, again and again through the night. In that rhythm, the hippocampus and cortex synchronize. Information passes from temporary to permanent. The wave does not know what it is carrying. It carries it anyway. That is enough. The brain that was worried today is the same brain that will consolidate that worry tonight. Strip some of its charge through REM processing, and pass the information forward with less of the original alarm attached. Sleep does not erase what was hard. It does something quieter, something more lasting and more useful than erasure. Time feels different in sleep. A dream that felt long may have lasted minutes. A memory replayed by the hippocampus in a millisecond burst may have taken an hour to form. The sleeping brain works at a speed the waking mind cannot perceive. What it does between midnight and 4 in the morning cannot be watched. It can only be allowed. The prefrontal cortex is last to fall into deep sleep and first to feel the effects of its deprivation. The judgment, the perspective, the quiet capacity to see the situation clearly. These restore during sleep. Each morning, they return as a dividend of the night before, more available than they were when the eyes finally closed. Every cell in the brain requires glucose and oxygen. During sleep, the brain reduces its demand, but does not eliminate it. The heart continues. The lungs continue. The brain continues, not consuming at waking intensity, but maintaining the conditions for the work it is doing. The body supports the brain even while the brain supports the body. There is no hierarchy. There is only the system. Could the sleeping brain actually renew itself, night after night, in ways that matter for a lifetime? It already has. In every person who has woken after deep sleep and found the problem clearer, the grief slightly lighter, the path more visible than it was the night before. The renewal is not dramatic. It is cumulative. It is quiet. It works. The brain you bring to sleep tonight is not the same brain that will wake tomorrow. It will have been edited, refined, cleared. Some connections stronger, some weaker, some new ones formed in the processing of today's experience. The change is subtle each night. Over a lifetime, it is everything. The fear of forgetting is partly the fear of losing the self. Memory is how the self coheres across time. But sleep is not where memories are lost. It is where they are consolidated into the form that persists. What sleep releases was already fading. What it keeps deliberately, carefully, and for reasons the waking mind will not always understand. The brain at 3 a.m. is not broken. It is busy. The window is narrow and the work is specific, and the conditions do not occur at any other time of day. 3 a.m. in a body that has been allowed to rest long enough is when the deepest maintenance runs. This is what the ceiling watching hours could not see. Adenosine clears during sleep. The pressure that accumulated all day, the weight of being awake, of thinking, of reacting, dissolves through the night. By morning, the brain is not merely rested, it has been reset. The sensation of waking after truly good sleep is not imagination. It is chemistry, working exactly as it was built to work. Every wave of slow oscillation that passed through the cortex tonight was the brain working. Every sleep spindle was the brain protecting its own work. Every REM cycle was the brain integrating what it has been given. The night was not empty. It was full of maintenance, full of care, full of the quiet industry of a system doing what it was always meant to do. The brain's capacity for change does not require effort. It requires conditions. Sleep is the most foundational of those conditions. Everything else, the learning, the experience, the practice, waits to be consolidated into permanence by the night that follows. The night is not the end of the day. It is what the day was building toward. Somewhere in the brainstem, a circuit older than language generates the rhythm of sleep. It has been doing this since long before there were words to name what it does. It does not need to be told to begin. It begins whenever you are still long enough. Whenever the day releases its hold and the body finally agrees to rest. The glymphatic system does not ask permission. When the conditions are right, body horizontal, brain in slow wave, channels wide, it begins its work. It does not care about the difficulty of the day or the weight of the week. It does what it was built to do, quietly, reliably, every single night that you allow it to. The brain that feared its own decline has been repairing itself throughout this hour. Not because of the facts, because of the sleep stages that have been gently deepening as the facts have grown quieter and the voice has slowed. The science and the experience are, tonight, the same thing happening at the same time. The real answer was never only about memory or toxin clearance or slow oscillations. It was about trust. Trust that the brain, given what it needs, does what it was built to do. That the night is not an interruption of the work. That the night is the work. That what you are doing right now, this breathing, this stillness, this letting go, is exactly enough. 100 facts ago, the question was what the brain does in its deepest sleep. Now we know it doesn't wait for the question to be asked. The maintenance begins the moment consciousness steps aside. We followed the sharp wave ripple from its origin in the hippocampus to the cortex that receives it. We watched norepinephrine reach its quietest point in the long dark of REM, and emotional memory arriving there with less alarm than it left with. What began as a fear of decline became something closer to a map of repair. The night was never empty. It was always, in its own precise and unhurried way, at work. The brain's process of clearance and consolidation is not something that happens to you. It happens for you, every night, in the only window when it can. What you carry into sleep is not what you carry out of it. Tonight, that difference is already underway. In these final minutes, take the deepest breath you can, as deep as possible. Think about consciousness itself, yours, and every other mind that has ever rested in the dark, allowing the same ancient maintenance to run. You are not separate from that. You are part of the long, quiet chain of minds that have slept, and repaired, and woken, and continued. Let your entire being dissolve into the ocean of rest, into the space before thought, before worry, before the question, into the slow oscillation, into the clearance, into the care the brain gives itself in the dark. Tonight, if the science of the sleeping brain brought you a quieter mind, a sense that the night is working rather than simply passing, a gentle like, subscribe, or share helps another restless mind find its way here. One quiet tap, one sleepy soul at a time. But there's no need to do anything now. Nothing at all. You've traveled far tonight. You can allow these thoughts of the rewiring brain to fade now, like the last slow oscillation passing through before dawn, leaving only the quiet behind. Let your breathing deepen. Let your body feel heavier, as if the neurons themselves have finally released their charge and the whole system has gone still. If sleep comes, let it carry you like the glymphatic tide, smoothly, inevitably, into a place where time and weight no longer matter. The Deep Sleep with Mind Science playlist has been following this question across many hours. The space where the brain's science and the body's need for rest meet. The next hour follows the part of tonight's question that this one didn't have time to finish. That hour is waiting whenever you are ready. But if your eyes are closed, stay there. Stay in the darkness that feels safe and warm. Tomorrow, the rewiring will still be happening. But tonight belongs to stillness. Rest well, quiet one. And remember, the brain, given the night it needs, does the work it was always built to do, where science becomes the softest place to rest. Good night.